Welcome everybody to our newest edition of the Spatial Biology Masterclass. Today we have with us Janusz Franco-Barazza. He's a research assistant professor at the Cookerman Lab and manager at the Spatial Immunoproteomics facility at Fox Chase Cancer Center in Philadelphia. Today he will talk about how he's using high-plex IF microscopy with Visiopharm's Phenoplex to generate spatial distribution data, identifying potential tumor-promoting or suppressive neighborhoods. And with that, I give over to you, Janusz.
Thank you. And I have to say it's a pleasure to be here again, sharing the steps that we are following to start analyzing this microenvironment in the tumor, which is the hot topic in biology now. And let me start sharing my screen. All right. Well, as Bettina already mentioned, the title. So yeah, basically, I want to introduce you guys the host factor and how we are planning to use it for screening and scoring the stroma of solid tumors. In particular, we're working right now with pancreatic cancer. But let me start with the details. So first of all, I have no financial relationships to disclose, but I'm a consultant of alpha and beta products for Luna4 Technologies and, of course, for Visiopharm. So I would like to start talking about the Paget's seed and soil theory, which basically was telling us that in order for cancer cells to thrive, they need to be in the right microenvironment, hence the right soil for this seed to grow. So we know the normal stroma has natural tumor-suppressive properties, and everything goes in balance until it's not. And when that happens, this soil becomes very fertile for cancer cells. And then when we can call it tumor-promoting stroma. Well, our duty in research is to understand how is this happening to provide the clinicians with the right tools, the right targets that we can attack either to prevent this from happening and never let the soil to become fertile, or the opposite, when we have already cases when the stroma is tumor promoting, how to tune it back to turn it into a tumor suppressive stroma. Well, so in order to understand that, we are working with one of the most, I will say, stromal-rich tumors, which is pancreatic cancer. And as you can see in this image, it's characterized by a dense fibrotic stroma, which is pictured here in teal, which is surrounding the cancer cells, pictured here in yellow. And as you can see, the ratio is overwhelming between stromal cells versus actual cancer cells. So imagine now that if this tumor microenvironment is fertile, how these cancer cells will be thriving. Well, in order to understand these dynamics, we are focused on studying the cancer-associated fibroblasts, which is the major stromal component of this microenvironment, but not only the cells, also their own extracellular matrix, which in turn, they form the CAF units. And this is how we are studying the biology of these entities. Needless to say, the pancreatic cancer is a rising threat and has been predicted to become the second deadliest cancer by 2026. So therefore, we have to really pay attention to this malignancy and conduct our research to prevent this from happening. Our lab, as I mentioned, has been focused on understanding the CAF units since several years ago. So we have been studying the biology from different aspects. And what I want to show you right now is a summary of some of the discoveries that we have made and how we are using those now to practically understand better the tumor microenvironment. So what you can see here is our beginnings doing multiplex staining. So I remember at that time we were having probably 7 or 8 markers. That was the maximum that we were able to detect. And with a very simple yet efficient code that you have here the link to. So we were able to start gating amongst the cell populations based on the expression of markers. This is the early times of what later Visiopharm is able to provide us. Okay, at that time, we started to understand that the normal microenvironment on, possess certain characteristics as opposed as the tumor-associated microenvironment. And amongst the tumor-associated microenvironment, we found that we can recognize two different phenotypes, the tumor-suppressive or the tumor-promoting. And in this case, just pay attention to the intensity of this marker, this alpha-5 beta-1 integrin in green, which is overwhelming appearing in the tumor promoting versus the tumor suppressive. So at that time, we already understood that the tumor microenvironment of pancreatic cancer had an increased organization of the ECM fibers, also called anisotropy. We detected augmented TGF-beta signaling in the fibroblasts of these environments. And we also found particular CAF extracellular matrix interactions, these structures that were having a particular signature. So we corroborate that these are clinically relevant because we found that when we were having this tumor promoting phenotype more abundant, so retrospectively, our patients were perishing faster than those that didn't have this particular phenotype. Well, our research continue, and now we also pay attention to the characteristics of the cytoskeleton of these cancer-associated fibroblasts. We found, for example, the expression of particular actin remodelers as pallidin and particular isoforms like 3 and 4. And along the talk, I'm going to be referring to the isoform 3, which is the one that we found is highly expressed in a tumor-promoting microenvironment. Recently, we also found the expression of unique proteins that normally are expressed in neurons. And it was a surprise to find them in the cancer-associated fibroblasts of pancreatic cancer. This is the case of netrin G1. And we found that the stroma of pancreatic cancer patients with high levels, as you can see here, of netrin G1 were diseasing faster than those that had less levels. Also, we found that these cells can secrete extracellular vesicles, and the profile of these extracellular vesicles is very unique. They normally have low levels of alpha-5 beta-1 integrin in the surface and high levels of netrin G1. So combining all these features, we came with a simple yet comprehensive signature to define what is a tumor promoting versus a tumor suppressive functional status in cancer-associated fibroblasts. And just as a summary, I want to show you the tumor promoting CAF have high levels of anisotropy, have high levels of TGF-beta signaling, increased 3D adhesions, a high expression of actin remodelers, and a unique profile with low targets in the unique EVs that we are having, that I just mentioned. But then we came with the question, okay, so these are basic findings. How can we put these to work for our patients? Can we combine all of them and monitor the stroma of these patients? So at this moment, it was a fortune to find, for example, Dr. Joshua Meyer, a clinician which is working in the radiation oncology department, and discussing with them the new therapies that they are using. We found that he discovered that by using a particular modality of radiotherapy that is called pulse low dose radiation. So the patients apparently were having less side effects. And how is this connecting with us? Well, let me first explain. Low dose radiation basically is the same dose that that patients are receiving for radiotherapy, but instead of being delivered in a single beam continuously, is being fractionated in micro doses and then is being delivered in pulses. So between each pulse, there is a resting period. So at the end, the patient is receiving the full dose that is supposed to receive, but as I said, in pulses. What is this causing? Well, this triggers a particular biological response where cancer cells, they still become radio sensitized, so they are dying, but cells that are not cancerous, hence, for example, the tumor microenvironment cells, have a chance to recover from the insult given by the radiation. And this was relevant for us because, as I said, this causes less toxicity in those organs that are radiosensitive. And it happens that the most radiosensitive organs are exactly in the gastrointestinal tract as cancer, as pancreas is. And as you can see, this microphotography, a very important player is TGF-beta, which is triggered. We know this normally with radiation, cells start to produce TGF-beta, but when the tissues are receiving the radiation impulses, the levels are very minimal or none. So why is this relevant? Well, TGF-beta, it happens to be a major driver of the fibrosis that is happening in the microenvironment of pancreatic cancer. So if we can tame the production of TGF-beta or the signaling, so we have chances to modulate the stroma from tumor-promoting to tumor-suppressive. And yes, exactly that is the question that we pose. So is PLDR able to induce a tumor-suppressive stroma, or still is gonna induce a tumor-promoting stroma? Remember, this is done and in tissues that are normal tissues, are radiosensitive, but are normal tissues. Now the question goes directly to pancreatic cancer lesions. For that, we started by studying the CAF units in a model that we have standardized in the lab since several years ago. I'm not going into details of those, but you can find more information following these references. So basically, we are growing tissue-derived fibroblasts that have been extracted from fresh biopsies from patients. We put them to grow and we allow them to construct their own CAF units. Once we have these 3D cultures, now these are radiated with a combination of gemcitabine and either the continuous radiation or the pulse low dose radiation. This is aiming to mimic the total neoadjuvant therapy that patients are receiving in the clinic. Once these cultures have been treated with these variations of radiotherapy, so now again, we can start questioning the presence of these markers to start understanding better the cell responses. And then after that, we remove the cells and we only keep the bioscaffolds, the extracellular matrices. Now we bring naive fibroblasts to evaluate the responses they are having to this alter in favor of the patient or in detriment of the patient matrices produced. So, but let's see the result that we found. So what you're seeing here is a staining of the extracellular matrix in these cultures. And the color coding that you see here is basically each fiber is color coded based on the angle of orientation. As you can see here, when these cultures are irradiated with the continuous radiotherapy, you start seeing more of the cyan color, meaning there is less variation in the tones, meaning the angles are mostly following a certain degree. The degree is not important. What is important here is that most of the fibers are aligned, are dense. Imagine these structures is denser and it gives more resistance than a environment that is a little less lax. Okay, so as I said, we found that we have an increase in the anisotropy when we are giving the radiotherapy, the continuous radiotherapy. But when we are giving these cells the pulse low dose radiation, so we see a decrease in this alignment, as you can see here. Well, we took then the effects of the conventional radiotherapy as our standard to normalize our data and to start seeing variations when we are just giving the gemcitabine versus the combination with the pulse low dose radiation. And as you can see here, when we follow either the TGF-beta signaling, the 3D adhesion structures, the actin remodelers that I mentioned, or the presence of the unique EVs, in all cases, although we didn't see a significant variation, all of them are following the trends that are coinciding with a tumor suppressor phenotype. Well, the next, as I mentioned, when we have these cells, we decellularize these matrices, and now we tested these scaffolds, which, by the way, as I mentioned, we are continue having the three-dimensionality, so we are mimicking the environments that the cells will encounter when they are seated. And with this technique that we already did it, so we know that the matrices can either trigger the activation of fibroblasts to become from quiescent to active fibroblasts, or if they are already active fibroblasts, depending on the matrix, this can sustain this phenotype or can revert it. So, and these are the results that we found. So we took two controls, the bona fide tumor-promoting extracellular matrix and a prototype that we have as a tumor-suppressive extracellular matrix. These are matrices coming from an alpha-B integrin knockout, and you can find more information in this reference. But basically, when we follow one of the activation markers as alpha-SMA, we see that the tumor-promoting extracellular matrix induce immediately the expression of this marker. And when the matrices are having a different phenotype, these are not able to sustain the tumor-promoting phenotype that these guys were having. Now, comparing this with the effect of the different type of radiation, we see that the combination of gemcitabine with radiotherapy, yes, indeed, is activating or sustaining, actually, sustaining the phenotype. But when we are providing matrices that were produced under the pulse low-dose radiation, these cells are having a decrease in the expression of these activation markers. So this was a biological proof for us that, yes, indeed, this type of treatment was causing an alteration in pro of the phenotype that we want to sustain in the patients to prevent the progression of the disease. But since we didn't have clear significance differences, so we went back to our data, we scratched our heads, and we start to try to understand how can we put everything together without paying attention to single parameters, now to understand in a more holistic way the biology of the cell per se. And for that, we run an analysis of the Z-score variations of all these trends showing here and put everything together as a comprehensive multi-parametric type of analysis. And that's how we came with the harmonized outputs of stromal traits or host. And now, since we can quantify this, now we are aiming to start scoring the stroma of this particular disease and extend this to other cancers to evaluate its importance. And I want to show you that when combining all these features and putting everything together, we clearly see the effect of the PLDR in comparison with the conventional radiotherapy or just gemcitabine. Well, this was very exciting, actually, but yet this is still being at the level of basic science, right? So we discussed our data with our clinicians. And before moving that, I just want to summarize what I just said, that by using our ex vivo model in a total neoadjuvant therapy modality using PLDR, we can achieve a low host factor features in the fibroblasts exposed to this treatment. Well, we again talked to Dr. Mayer, and we put together a clinical trial in-house, which was practically focusing on assess the efficacy of radiotherapy escalation. Since we saw that this type of modality apparently was not altering much or aggravating, I should say, aggravating the phenotype of the tumor-associated fibroblasts. So now we're there to start comparing the regular dose the patients are receiving with a little higher radiotherapy that indeed will could help us to tackle the cancer cell without affecting the stromal cells. And basically is what I just described in vitro. We are trying to mimic something similar with the participation of patients and then assessing exactly the same parameters that I just showed you. And this is when we came with the new technology that is now available. And here I'm just talking about one of the instruments out there that could perform this type of technologies. Then we can now, through a multiplex single cell analysis, we can understand better the stroma of these cancers. And the first thing that you might think when you see these colorful pictures, at least in my case, is that we are seeing a star at night, isn't it? So, and here is when we start to have blurry lines between what is a biologist and what is an astronomer, because despite the fact that we both are using sophisticated equipment, I believe the difference is that instead of looking at the microcosmos, we are looking at the at the macrocosmos. We are looking now at the microcosmos, and indeed. So following some of the principles that studying celestial bodies that are followed, we can also understand what is going on in the tumor microenvironment. Because, for example, when we analyze the Pinwheel galaxy, we know exactly that it's composed of several type of lights, as you can see here, and everything composed giving us these beautiful images. Well, something similar is happening with these multiplex images that we are analyzing. So where we have different signals that when they are put all together, they not only give us a beautiful image, they are providing a lot of information that now we can extract in a single cell matter to understand exactly the interaction of these cells and the phenotype that these guys are having when they are close to each other or away, etc. But again, so these signals come with a lot of noise, and we need to clean them up before starting to analyze them. So here, I'm just going to show you a few of the steps that we follow in these examples when we need to clean one of the biggest problems that we find when we are working with immunofluorescence, which is the natural autofluorescence of the tissue. And as you can see here, after removal, the images come clear. So now we can see the two markers clearly and the distribution within the cell. But the next step that we are following is also taking the background that the secondary antibodies could live there to also try to prevent false positives in our signals. As you can see here, we have already removed the autofluorescence, but we haven't removed the background of secondary antibodies. And after that, we can get cleaner and more trustable images. So after these processes, now we move to the analysis that is conducted in Visiopharm. And I want to show you some of the characteristics of this pipeline called Phenoplex that working together with Visiopharm, we have been either learning how to use the power of it, but also suggesting what things are needed or what things could be better in order to improve this tool. So going back to an image that contains a lot of signals, I'm focusing on a particular neighborhood that I am highlighting here. We can now focus our attention on the cells that are within this area. And Phenoplex, one of the beauty of this pipeline is that we can recognize each single cell just by the presence of its nucleus. And based on that, we can not only detect, but also segment the cells that we are seeing. You now can see cells in blue with a white hallus that I'm going to show you. This is a single area, but imagine a full tissue when we can have thousands of cells from where we can obtain very valuable information. And going back to the segmentation that I just described, is beautiful how we can discriminate the signal that is only in the nucleus or in the cytoplasm or in both. So this is now giving us the huge advantage, not only to detect the presence of regular biomarkers, but also to monitor the activity of certain proteins, since we can identify phosphorylated or other type of post-translational modification of these cells, of these proteins, if they are changing the localization within the cell. So with that level of detail, now we can start understanding better the functional status of these cells. Well, so now going back to the image, the cells that were already detected and segmented, now they are entities within the area of study. And within this area, we can start querying what are the characteristics of each of these cells. For example, what I was doing here is just checking which are the pancytokeratin cells. And exactly we can see the distribution of those versus CD68 positives or CD3s or CD8s. As you can see, it does a great work detecting each of them. Well, having this powerful tool, now we went back to our clinical trial. And during the time that we were harnessing the power of Visiofarm, we were continue selecting and acquiring patients. And as you can see, trying to match also controls based on sex, age, and race to make a stronger call from this data. Well, during the time that tissues were ready, so we developed an internal pipeline to study the host positive CAF cells in pancreatic cancer. And here I want to spend most of the time because although this is a pilot, I think this is a very strong approach that we are taking, and I hope I can convince you that this is going to render very important information. So taking a TMA, so base the analysis on two different cores, basically a normal tissue versus a bona fide pancreatic cancer tissue. So we conducted a multiplex staining. Here I'm just showing you a few colors just to identify the cells by the nucleus, the presence of pancytokeratins, immune cells by the presence of CD45, and fibroblastic cells by the presence of vimentin. You can see immediately the architecture, how different it is between a normal pancreas and pancreatic ductal adenocarcinoma. And what we did is now based on the same autofluorescence that I was mentioning that we need to clear in order to do the analysis. So we also use that particular property of the tissue to identify in the image in a non-biased and automated way. Where is the tissue? It's called the tissue detection and is within the pipeline that was developed for this type of analysis. Now, since we are practically gating the analysis only to the area within the yellow perimeter, now we can detect and segment all the cells. And for example, here we are obtaining a certain amount of cells from which we are conducting the analysis. Well, the first thing that we did is to check the expression of pancytokeratins, vimentin, but from the perspective of the singular identities. And here we can immediately verify the accuracy of this pipeline. Since pancreatic cancer is well known to have a very strong difference in the amount of fibroblastic cells versus epithelial cells. And as you can see here, practically the ratio is totally inverted. We have this amount of of epithelial cells versus this amount of fibroblastic cells. And when we look at the normal tissue, this ratio is completely inverted. This is the regular or the biological, physiological, actually, architecture of the tissue. Well, knowing that this pipeline was working as expected, we started to gate the populations to start separating these cells. For example, we started to check the presence of pi-cytokeratin, but in the absence of pigmentis, to start detecting only epithelial cells, either normal or cancerous. The same we did for immune cells. We did in the absence of pancytokeratin. And when we went to assess the fibroblastic compartment, we checked the presence of the marker pimentin in the absence of the former. So as you can see here in the inserts, this is the raw population, and then in the magnification, after cleaning or after gating the population, how can we be more assertive in assessing the cells that we are interested in? Well, focus on this fibroblastic population. We went to check for the presence of the markers that I just mentioned at the beginning of the talk. And to our surprise, I started to check the co-expression of all these several markers to understand better which are the markers that are more relevant. As you can see here, when I started to check these particular markers, either phosphosmad, pallidin, phospho, etc., I barely or practically I didn't see any co-expression. But then, based on our previous research, we know, for example, that netrin G1 and phosphofac, they correlate in the co-expression when the disease is progressing from the tumor adjacent fibroblast to the bona fide tumor associated fibroblast. So I started now to cross those parameters, and then is when we found the correlation that I was mentioning. So based on this correlation that I was looking, we came with this signature that is composed of these particular markers, in which now we included FAP. So then we went to look exactly for this particular subpopulation of calves. And this is what we found. As expected, the normal tissue has a practically minimal amount of these cells present in the stroma of this tissue. On the contrary, pancreatic cancer has a very, I will say, important amount of cells distributed in this tissue. And then we went to see exactly where are they located using one of the tools that is embedded in the Phenoplex study of Visiopharm, which is the spatial analysis. And basically, I delimit my study to start checking what are the cells around when taking a target cells, which are the cells in the periphery no further than 10 microns. And this is exactly what I found. So I was expecting to see this terrible quadruple positive fibroblast supporting the tumor, surrounding the tumor. And to my surprise, you can see here in yellow, few cells are surrounding the epithelial cells in this particular case. As you can see here, this is the mean of cells that are practically in the periphery of 10 microns from epithelial cells. So it's a very small number. But when we look now about immune cells and host-positive calves, so we now know that for every immune cell, we have, sorry, the opposite. For every host-positive calf, we have 3 immune cells nearby, suggesting that this particular subpopulation is interacting mostly with immune cells rather than with the cancer cells. So our attention went there, and just out of curiosity, I started to monitor from the origin of the cell to 30 microns away, what is the distance in where it's more common to find these cells? And well, as expected, so epithelial cells and these particular calves are far away, but they are closer to immune cells, as you can see here. Well, continue with the scrutiny of the neighborhoods where these host-positive calves are. So I question, out of the immune cells, tell me now what are the the T cytotoxic cells, and still it's a fairly large number of cytotoxic cells nearby these fibroblasts. But interestingly, when starting to look for activation marker of these cytotoxic cells, for example, proliferation, Ki67 positive, a very small number of cells are proliferating when they are close to the host-positive calves. Then I say, well, okay, so this suggests a tumor, an immunosuppressive environment. So I went to check for the expression of PD-1, and indeed I found that roughly 1 in every 2 immune cells, this is cytotoxic near to these fibroblasts, are expressing PD-1. So I went to look further, and what I catch my attention is that looking to B cells, helpers, or cytotoxic, which are the 3 major population of immune cells that are surrounding the host-positive calves. So these cells are not proliferating. These cells are producing, half of this population are already producing PD-1, and in particular, for example, T cytotoxic, a very small number are bona fidely active. So this is really, and by the way, dendritic cells were also close to these cells. But again, when checking the proliferation of these cells, it's very small number. So again, suggesting that these host-positive fibroblasts are practically, could be sustaining a tumor-promoting calf, since they are practically surrounded by cells that are immunosuppressed. And with this, I want to leave you some take-home messages. First of all, we found an increased prevalence of these host-positive tumor-promoting calves in the specimen of pancreatic cancer versus normal cancer. These fibroblasts are interacting mainly with B cells, T helper cells, and T cytotoxic. But all of these are, they seem to be immunosuppressed. And with this, I just want to close saying that the spatial proteomics profiling now is offering new translation opportunities, because now we can make more comprehensive analysis of the tumor microenvironment. And we, in particular, we're planning to apply this knowledge, combining now the functional status of the tumor-promoting calves with the interaction of immune cells. And I want to close just showing you Edna Zuckerman Lab and the people involved. I want to acknowledge all of them, in particular Tiffany and Christopher, who did the in vitro assessments that I showed you at the beginning. Our clinical team, Dr. Joshua Mayer, Sanjay Reddy, the facilities, and of course, we couldn't start doing this without the help of Visiopharm, in particular, Fabian and the Rasmuses, who were working very closely with me to trying to understand and use these tools to generate the data that I show you today. And hopefully, I would like to catch up with you guys in the next cycle of master classes, now showing you the data that we compile from the actual clinical trial using these tools. With this, I'm closing here, and I will be happy to answer the questions that you guys have.
Wow, thank you. That was really very insightful. Very cool presentation. So we already have the first questions coming in. So there were several ones from Fabian. We'll just do them one by one, I think. Is the process of irradiating the calf similar to the radiation of fibroblast feed layers for embryonic stem cells?
So the continuous radiation, yes, is similar, but the pulse, it differs exactly on what I just said. It's coming in pulses, and that allows the cells not to die, but to recover, which is the opposite that is the goal of irradiating the fibroblastic feed layers when you want to get rid of them.
Okay. Does this inhibit their proliferative capacity?
That's a great question. We are about to start checking that. I can tell you from the analysis in vitro, we didn't see a substantial decrease in proliferation, nor in dead cell when we are using the pulse radiation. So I don't have the numbers in hand, but I can tell you that is just by eye, no, it's not, doesn't seem to be affecting proliferation.
Can you also induce the phenotype with non-cancer tissue fibroblasts?
Well, yes, it can be. Yes, actually, that's what our goal. Try to understand how can we induce the tumor suppressive phenotype. So instead of getting rid of the tumor promoting fibroblasts, just tune down the phenotype. And this is from experience that we have the community when trying to get rid of calves from pancreatic cancer. So clinical trials have failed because these cells are needed, but they just need to be well behaved, not supporting the tumor.
That sounds good. Mats, at the start of your talk, you showed the stretched alpha SMA networks. Is there a possibility that a non-fertile microenvironment uses these networks to physically block cancer cell movement or even angiogenesis and thereby nutrient-starved cancer cells?
Yes. That's a very good point. So let's keep in mind, by saying cancer-associated fibroblasts, we are talking a large population composed of several subpopulations. And yes, we have subpopulations that are expressing heavily alpha SMA and other proteins as FAP. And these are called the myofibroblastic subpopulation that, again, it can be subdivided. But yes, those things, these guys have also a very strong extracellular matrix component in the genetic transcription profile they have. So yes, indeed, they are depositing more extracellular matrix. They are making the environment stiffer and not accessible to other immune cells, and even in some cases also collapsing the vessels, so making the nutrients internalization more complicated. And then switching to other mechanisms that I will take even longer to explain right now. But yes, it definitely are changing the tumor microenvironment in that way. Yes.
Okay, that sounds cool. And here another question. Have you looked if TP-CAF cells are promoting the survival or expansion of Tregs? Are they in proximity?
That's a great question. We are definitely poised to start checking that. In the preliminary examples that I show you, I know about the T helpers, and out of that, I want to know the Tregs. What is the status of those? Because you're right, it makes sense if it's an immune suppressed environment. So chances are we have more Tregs there, right?
Okay, and there's a question from Susan. Nice talk. Have you quantified the ratio of TP-CAFs and TS-CAFs in PDC TME? Thanks.
No, we haven't yet, but that's one of the things exactly what we want to check in the patients. Remember, this was just a pilot. in 2 samples. But now that we have completed the accrual of the participants, yes, I'm eager to start checking these tissues indeed.
So there's more data to come that we can show later on.
Absolutely. Yes, absolutely.
And there's another question from Fabian. Are the CAF populations in immune invasive and immune excluded PDAC different?
There should be. Yes, there should be. That's part of the deeper analysis that we definitely want to conduct now that we have the right tools to do so. Yeah.
Cool. There's another question. Can this analysis process be in 3D form?
Oh, that's my dream, indeed, to be able to analyze the tissue samples in 3D. We are not there yet. I think the technology is starting to evolve and moving to that scenario. We are still working in 2D, but nevertheless, this is not a small thing because we can already understand in X and Y the distribution of these cells. Now, if we combine several consecutive cuts, we can more or less reconstruct the 3D environment and tracking the cells in these several consecutive cuts. There are now digital technologies that could allow us to put everything together and indeed start correlating the presence of markers in 1 cut versus, I don't know, 5 cuts later, trying now to put everything in a virtual reconstruction, 3D reconstruction.
Cool. And the next question, were there any surprises coming from the spatial neighborhood analysis that you found, which you would not have found without neighborhoods?
Well, definitely. So as I mentioned, I was expecting my my host positive CAFs to be very close to cancer cells, helping them to survive, etc. Because there are several aspects. One of them that I didn't mention is that in collaboration with other groups, we have found that when nutrients are scarce in the tumor microenvironment of pancreatic cancer, are actually fibroblasts feeding cancer cells by transmitting nutrients from cell to cell all the way to cancer cells. So my expectation was to find them there. And then to my surprise is no, they are distributed in different regions and without understanding the distance, the localization, I couldn't have guessed that, yes.
Okay, that's fascinating. Not seeing any additional question right now. Then I think we're through with the questions. Thank you so much for being here and sharing this with us. And looking forward to a follow-up webinar once you have the data. That's
absolutely
very
yes.
interesting. And
absolutely.
Yeah, we see everybody.
Thank you everyone for attending, and thank you Visiopharm for the opportunity to showcase my research. Have a good one. Bye-bye.
Bye-bye, everybody.